So next, we will welcome our second speaker of the Young Scientist presentation, Dr. Siew Jian Jing, who's doing a postdoc research fellowship at the Academia Sinica in Taiwan. So he's doing a neuroscience, and he will bring us through quite a different human organ now, the brain now, and talk about microglia and sugar-binding proteins in neurodegenerative diseases like Huntington's disease. Please, Dr. Siew.
Good morning. My name is Siew Jian Jing, and I work in Academia Sinica Taiwan as a postdoctoral researcher. And today is truly my honor to present you my story about microglia and the sugar-binding protein in neurodegenerative disease. So I have been working with the neurodegenerative disease such as Huntington disease that suffer from this kind of movement disorder, as well as tauopathies such as Alzheimer disease that suffer from this memory impairment. And up until now, there's still no effective treatment that is currently available. So, talk about the brain. Inside of the brain, there are 4 different types of major cells, which include the neuron, the oligodendrocyte, astrocyte, and the microglia. And in neurodegenerative disease, there will be like degenerations of this neuron and the activations of other type of glial cells, and particularly this microglia. So what is a microglia? Put it to the simple word, microglia is the immune cells of the brain. And let's look at these pictures. Don't be afraid about this. Look at this one. This red color is a marker for microglia. So whenever you see these red colors indicate those are the microglia cells. And here you got this R62. This is the model for Huntington disease. And like Professor Tim Han said yesterday that as a biologist, we like control, right? So with this R62, this is the disease model. Then we have this wild type. Wild type is the control, means that these are the healthy mice at the same age. So as compared to the wild type, you see this R62 microglia, they have these green signals, which call this galactin-3. So this galactin-3 is the sugar-binding protein that we mentioned. They particularly like these beta-galactosidase sugars. So, interestingly, in the normal or healthy brain, the level of galactin-3 is very low or just absent. However, in Huntington disease, this protein is being upregulated and is also a sign for the microglia activations. So, if we look further into this microglia, we found that a portion of galactin-3, actually this red signal, now the red signal is the galactin-3, they are actually colocalized with lysosomes. So you can see here, inside of these lysosome structures, some of them, they have this galactin-3 puncta. So what it does that is that we found the accumulations of galactin-3 in this damaged lysosome, they actually interfere with the clearance of these damaged lysosomes. And the implication of this is that it causes the dysregulated inflammatory response contributed by the microglia. And interestingly, if we inhibit galactin-3, no matter by genetic suppressions or pharmacological tools, we can effectively promote the clearance of the damaged lysosomes. And this eventually reduces the inflammatory response triggered by the microglia. These are just the cells and the mouse model we have been using to prove these concepts. So now it's still at the cells level. How about in the mouse model levels? Remember just now, we look at the Huntington disease patient, they have this movement disorder. So here, this is the assay called Rotarod. It can be used to measure the movement capability of the animals. So this one is the R62 mouse model, means the one with the Huntington disease. So you can see here, now is the movement. Compared to the one with the galactin-3 inhibition treatments, you can see that it performs significantly better compared to this mouse, which is the Huntington disease mouse model. So from here, we can chart the result and found that galactin-3 inhibitions significantly improve the motor functions, the movement of this mouse model. We have completed this study and get it published, so for the further details, you can look at that. And next, we actually move on to the second part, which is the tauopathy, such as Alzheimer's disease. So when we talk about Alzheimer's disease, it's like there are two hallmark proteins, including the hyperphosphorylated tau, as well as the amyloid beta plaque. And interestingly, in both the hyperphosphorylated tau and the amyloid beta plaque conditions, we get to see that there are significant upregulated signals of this galectin-3, the green color. So you can see here, when the green and the red, they appear together, they will show as a red color. And let's go back. One thing is interesting is that if you look at here, these white or gray color signals, those are the disease-associated protein. And one interesting observations we found that is that galectin-3 positive microglia, they only appear closer to those abnormal disease-associated proteins. So if you compare the microglia here and the microglia here, they might have different properties. And the cutting edge here is that we use the single-cell RNA sequencing so that we can compare the property of these cells to the property of these cells. So with that, we use this mouse model, and after the brain dissociation, we collected the microglia cells and subjected them to the single-cell RNA sequencing. And here, we are managed to identify 12 clusters of microglia. And from here, we can compare the microglia with or without the galectin-3 expressions. And here, we managed to identify those specific genes that are dysregulated in this galectin-3 associated microglia, or we call them the game genes. And what's interesting about this game gene is that a lot of genes being implicated here are actually the gene associated with Alzheimer's disease, which further proving that these galectin-3 associated microglia plays important role in Alzheimer's disease. So from here, we can perform the biological analysis using the gene ontologies, so we can predict what kind of functions being regulated by this gene. And here, these are the major biological processes regulated by game gene, including the inflammations, immune system, translation and ribosome, chemotaxis, protein folding, ATP, and apoptosis. Now we know that in this galectin-3 associated microglia or the game gene, the microglia has upregulated of these kind of functions. So how would it do if we remove galectin-3 from the Alzheimer's disease, the tauopathy mice? From here, we cross these tauopathy mice with the mice without galectin-3. And we can obtain the tauopathy mice without galectin-3. And here, we perform biochemical assay as well as behavior study, which include this learning and memory assay, and by using this Morris water maze. So what is a Morris water maze? It's the nature of the mice that they dislike water. So if you put a mouse into the tank filled with water, they will try to escape. And for the wild-type mice, after a certain period of training, they will remember there's a platform here. So the mice will locate this platform and stand here because they dislike water. Now, the next, I will show you the recording on how the mice will move in the wild-type conditions. And this video is fast forward. So this is the trail by the mouse. They will swim and they locate this platform. So after training, they remember here is the platform. Next, we look at those with the Alzheimer's disease and Alzheimer's disease without the galectin-3. Again, let's look at the trail. You can see that mice with galectin-3 removal, they remember where's the trail and they can locate the platform. But this one is still finding the... They thought, maybe he thought he remembered the platform. He either remember wrongly or he doesn't remember at all. He's still finding where is the platform. So with that, we can record this trail and then perform analysis. So from here, you can see that this black and the blue line means the wild type, the control mice, and this red color indicates the tauopathy mice. They use significantly more time to locate where is the platform, while the tauopathy mice without galectin-3, they can locate the platform much faster, means they remember. We can say that galectin-3 inhibitions prevents the learning and memory impairment in tauopathies. And for translation purpose, we also use this human iPS-derived microglia. And from here, we treated the cells with or without the hyperphosphorylated tau, the disease-associated protein in tauopathies, and then also with or without galectin-3 inhibitions. And then from here, after the RNA sequencings, we managed to identify what we call this galectin-3 associated microglia early responsive gene, or we call that the gamers genes. And it is also very excited to see that a lot of these gamer gene also being implicated in Alzheimer's disease. So for functional validation of this gamer, we treated the conditioned medium collected from the cells to the tauopathy neurons. And we assay how it will affect the misfolded tau. And from here, you can see, indeed, with the galectin-3 inhibitions, the pathogenic tau, the misfolded tau, show a decreased levels. So from here, we can really say that if we can suppress galectin-3, it will actually reduce the level of misfolded tau. And if we go further into what happened in the conditioned medium, this one I'm not going into all the details, but we can conclude to say that inside of the conditioned medium, there are more exosomes. And these exosomes are actually filled with a lot of disease-associated protein and galectin-3. So I will just go with this schematic diagram to explain the mechanisms. In the tauopathy conditions, the hyperphosphorylated tau released by the neurons, they will activate the microglia, become the galectin-3 associated microglia. And under such conditions, these microglia release a lot of abnormal exosomes. This exosome is just like a letter that can pass from one cells to the other cells. So at the recipient cells, when it receives this abnormal exosome, it will further escalate the disease condition in these recipient cells. And if we can suppress the level of galectin-3 on the microglia side, we can actually reduce and correct these exosomes, and then such that when the recipient cells receive these abnormal exosomes, it don't have the increased level of the misfolded tau. Of course, we also show that there are regulations of the free form of galectin-3 and pro-inflammatory cytokines, but it's not being included in these studies. And then we further move into this part and found that another property of galectin-3 that contributed to the misfolded tau. Under the normal conditions, the pathogenic tau, they will accumulate and become a lot of this misfolded tau. But in the presence of galectin-3, it can act like a molecular glue to promote the oligomerizations of this pathogenic tau and resulted even more misfolded tau. So that brings to my summaries. The accumulations of galectin-3 and damaged lysosome interfere with the clearance of damaged lysosomes. And galectin-3 promotes the production of exosome filled with disease-associated protein. And binding of galectin-3 with the pathogenic tau enhance the oligomerization of this pathogenic tau. Inhibitions of galectin-3 may serve as a new strategy for therapeutic intervention in neurodegenerative disease. And with that, I would like to thank all the people that have contributed to this project, and particularly Dr. Yi Zhuang, my supervisor, who has guided me throughout the years. Thank you, and I'll be happy to take questions.
Thank you, Dr. Siew. Now, Dr. Yang will be moderating the Q&A session as well.
Thank you, Dr. Siew. That was a very interesting talk. Please, sit. All right, any questions from the floor after we've heard about how we can target GAL3 towards neurological disease? Yes, please. Yes.
Hi, a very nice talk. So I was just wondering, I have a very short, technical question. So I was just wondering, I'm not sure if I missed it, but what's the direct mechanism through which galectin has its function? Because in your iPSC model, you did a RNA-seq after, in presence or absence, inhibiting galectin, but it's like a sugar-binding protein, so its direct effect probably comes from, adhesion or some other type of mechanism, not through transcription, presumably, right? So transcription would probably be a much more downstream effect, right? So the target genes you're looking at, so just wondering if you could draw a more direct mechanistic link. Does that make sense?
Yeah, thanks for the question. This is actually very important questions. Galectin-3 has multiple functions. It has been studied about 30 years ago. And when people start to look at galectin-3, they notice it is involved in the macrophage activations. So the other name of galectin-3, if you search through from the literature, is actually called the MAC2, the macrophage activating factors. However, besides of the activations of macrophage and inflammatory response regulations, in the recent years, people start to notice another very special functions of galectin-3, in which they bind this abnormal sugar on the vesicle. So inside of any type of intracellular vesicles, supposedly the sugar is on the intravesicular side. However, when there's any damage, let's say a leak of the sugars, it will get exposed. And galectin-3 can recognize this type of sugar and become the pangta like we showed just now. So this is something new and totally different concept. And from here, as you described just now, that you notice from the single cell RNA sequencing that we see that galectin-3 involved in a lot of inflammatory response, like and immune system. Those are like what we really already know. Then what we really found in the different way is that it also inhibit, when we bind with the sugars, it also inhibit the clearance of these damaged vesicles. These are something totally different. And on the second part, when we see that this galectin-3, they also can bind on those misfolded protein. And when bind with this misfolded protein, they kind of like aggregate them. This protein by itself, they have the property to aggregate, but if we add galectin-3, they even further aggregate. So these are all like the disease condition in the Alzheimer's disease. Thank you for the questions.
I did have another question. This is just out of curiosity, just out of interest in glycobiology. So, like most of the disease phenotypes that's related with sugars, like we often see that there are changes in sugar. So like in cancer, like you have phenotypes that are associated with changes in sugar molecules. So I'm just wondering, since galectin is a sugar binding protein, like are there like individuals who might have a different pattern of like expressing different sugars and more susceptible to Alzheimer's? Did you look at that?
This question is like, is still ongoing.
Right.
I have been working on this for like, on the sugar part, the specific pattern, but it's not easy actually.
Yeah, it's quite technically challenging. That's why I was like, yeah, we often like want to target like these sugar molecules, but there's like, you don't really have anything other than antibodies that can like specifically recognize, and you're just
I think the major obstacle is that even we manage to identify certain type of sugar pattern that happen in the neurodegeneration, even when we say neurodegeneration is a broad term, let's say Alzheimer's disease, it's very difficult to, at the moment, to pinpoint the function of this kind of sugar patterns. But I think with the advancement of technology and throughout international collaborations, we can get it, at least some clue.
Great, lovely, thank you.
Thank you, thank you. Does AI or machine learning help to identify that, or? It's okay. Just a general question.
I think the major thing is that currently the data available for the brain glyco is really limited. So I'm still not sure whether with this limited data we can train the AI.
I see, I see. Got it. Thank you. Yes, I know you've been waiting for a while.
Thank you. It was an amazing presentation. And you kind of already talked about a few of the future analysis, but I was wondering if you have plans to analyze the effects of this inhibition in other hallmarks of Alzheimer's disease, as the accumulation of the amyloid peptides and oxidative stress, neurotransmitter reduction.
Yeah, that's a great question. We focus on this hyperphosphorylated tau, but the other two groups has worked on the amyloid beta part, and they have very similar results as us.
Thank you, that's really good.
Thank you. Yes, please. Yeah.
Really interesting talk. So my question is like amyloid beta, so that is also a protein which aggregates and oligomerizes in Alzheimer's patients. So have you seen galectin-3 interacting with amyloid beta or increasing the aggregation propensity of A beta?
For the amyloid beta part is kind of straightforward, because the amyloid beta being released by the cells, and at the extracellular level, they bind with each other and become the aggregate. So one of the property of galectin-3, besides being recruited into the exosome, they also can be secreted in the free form. So that one is actually the free form direct binding with the extracellular amyloid beta and promote the aggregations.
I think galactin-3 interacts with...
Sorry?
Can you repeat?
So you think galactin-3 interacts with A beta?
Yes, that has been shown by the other group.
Thank you, thank you. Yes, please.
Thank you. What do you think could be the side effects of the, or did you have a side effect in the wild type knockout mice?
Yeah, that's the beauty about this work, because in the normal mice or healthy mice or young mice, you don't see galectin-3 in the brain. Only when we start to age or we have neurodegenerative disease or we actually bang our head, then the galectin-3 start to express on microglia. So from what we can observe or from the behavior analysis that we run, we do not see any side effect of galectin-3 inhibitions.
Thank you. Any more questions? If not, may I ask a very general one? So does it mean it's not preventive, but it will actually slow down the progression of the diseases rather than preventing it from happening at all?
I would say it's more on slowing down. And if we, for translation and the medical purpose, I believe it has to be combined with other drugs. Yeah.
I see. Then is it a matter of reducing the symptoms as well in targeting GAL3?
Yeah,
all right.
definitely.
All right. Thank you, thank you. Any more last questions? If not, due to time constraint, we will wrap up the session now. And now it's time for certificate giving.
Yes, thank you, Dr. Yang, and thank you, Dr. Siew.